Thanks to Valerie Paul and Joni Cleopas have been kind of the cornerstone of the program committee from the very start, and I'm extremely grateful for them and all the hard work they put in. And they raised a very good issue about, typically these meetings end with the last talk, the last beer, everybody goes home, and then you wait for 4 years to come back. And so it was their idea to say, can we do a little bit better and come together as a group to kind of review what happened. I don't know about you guys, but I was not able to see a fraction of the coral science talks that I wanted to see. Part of it was my day job, but I know a lot of you couldn't either. So this is kind of the cliff notes of the 13th ICRS. And at this point, I'd like to ask, is Joni going to do first? Joni? To come on up, and thanks to both Joni and Val for taking on the opportunity to have a good synthesis at the end. So thank you. Thanks, Sam.
Okay, I think what we really want to do is just build on the momentum that was really conveyed here through all the excellent plenaries, all the discussions. I think some of the biggest change I've noticed here is from the other previous ICRS meetings is really we are moving from doomsday and despair into, the we can do it kind of attitude. And so I really want to use this last session to sort of solidify that and give people some ideas or hear their ideas about how we move from this monitoring the decline of reefs to actually turning things around. And I think there's a lot of positive stuff in this meeting to talk about to do that. So the format for this, I don't know how to change the slides. So the format for this meeting, what we're going to do is have 3 different panels. On each panel is 6 scientists that are going to summarize for you some of the topics in this meeting that you probably didn't get to go to. So this, as Bob calls it, this is a really nice way to get the cliff notes. So they each get 5 minutes to summarize a world of information. It was a Herculean effort, particularly for those people that we contacted yesterday and asked them to do it. And then following that, we're going to have 15 minutes of panel discussion. And this is where you guys in the audience get to come up and ask the panelists questions that have to do with the science. So one thing we want to do is we want to make sure that this is mostly focuses on the science. This morning we talked a lot about the activism and the things that we can do to reduce CO2 emissions and so forth, and the other insults to reefs. This time we want to really concentrate on the science and where do we go as scientists to make this happen, make this action happen. But the first thing I want to do is I want everybody to stand up with your stuff, and there's a few things, and I know I'm going to be a little ahead of myself on this. But come up to the front. This is good. Yes, breathe, stomp your feet, whatever you got to do. So, we want to really get together a coral reef action plan. So give me a C. Yay. Give me an R. Give me an A. Give me a P. All right, that's our new acronym. Okay, I'm sure there's going to be a lot of accolades here for what Bob has done. I've watched him toil through this for months and months. He's done a remarkable job, and I think it's been a remarkable conference. So in case we don't have a time to do this, and there's plenty, we can always applaud him multiple times, but let's give Bob a real. Okay, so now we're going to get started. So I'm going to ask the 6 panelists for this first discussion to come up. And also, while they're coming up to the front, I'd like the speakers up here too. Really want to, what we told them is we want to know what the key findings are, what the key questions for the future research are, and what are the main points the ICRS members, you guys, should take away with you. So it was a hard task, and I really appreciate it. And I think Val is going to come up here and introduce the rest of the people. So thanks.
So what we've done is ask 3 of the panelists to give a short 5-minute summary of, we've asked them to cover quite a Herculean task to do that and summarize these different topics in the 5 minutes, but we'll go ahead and get started with that, and I think Mark Eakin is first.
Thank you. Let's see. Do I advance it up here? Yeah, there we go. So Terry Hughes and I will be representing climate change, warming, ocean acidification, and multiple stressors. Just a small topic. So one of the things that we have seen this year, and starting in 2014, is that we've seen the biggest and longest lasting bleaching event ever. It's completely changed the way that we look at these events because we've never had anything that lasts this long and has the impact that we've been seeing. At this point, it's even bigger than what happened in 1998. You see some statistics here that over 40% of the reefs have reached at least alert level 1. The alert level 2 area, meaning those reefs with severe widespread bleaching and significant mortality, is greater than the size of the state of Massachusetts, or about 50% larger than Wales. I could go on with a list of things. And over half of the places at any of these stress levels have been exposed twice. This has been a major event. And as you can see from the large areas of light blue that are the areas where there's no stress, this has been a widespread event. We've seen severe impacts. Places like Christmas Island, where they had 80% mortality, Jarvis, where they had 95% mortality. Some of these places were hit really hard. And we were able to see that, and on a large scale, these things are highly predictable. As we go on a finer scale, we see variability. I could have quoted Terry's talk, but instead I quoted a tweet of his from May, that corals declined by 50% on the Great Barrier Reef in the last 30 years. Bleaching did the same thing in the north in just 2 months. That far northern section that had previously not declined as a result of the stressors that had caused problems to the south. But there was a great variability on a spatial scale across the Great Barrier Reef, but it was, largely at a predictable level at that point. But one of the things as we start looking at the science of all of this, and we hear from the many speakers who looked at things in fine detail, and this is just one example from Tyler Smith's talk, that there's a lot of variability when you start looking at things like how deep are the colonies? What species are we looking at? We had talks that looked at fine-scale variability and saw things where the degree heating week pattern started to drop out and didn't really work. And the reason is because there's far more going on that you need to know about than just what's going on right now with bleaching stress or thermal stress. And that's the reason why we need to be doing the research that we're doing and the detailed studies to really understand the various stressors that are involved, how they've been affecting the corals in the past, and how they're affecting them today. And in fact, that past history really matters because some of these corals have been building up their resistance. I love this diagram by Noah Pomeroy. He was actually talking about hitting the thermal stress gem, but it really applies to all of the segments we're talking about. Some of these corals, just like the super corals that we heard Ruth talk about in her plenary earlier, are trying to get ready for these big events and hopefully will survive long enough to do so. And a lot of the science that's been going on has been looking at that. But we also have to look across multiple stressors. And we not only have to be looking in the lab, but we have to be looking out in the field because not only do these things interact in ways that we can study them in an experiment with crossing 2 factors or maybe 3 factors, but a lot of these things interact broadly. So in general, the climate change, the warming signal is that longest, most widespread bleaching event ever. The discussions, the presentations were all about the how and the questions of why. Do bleaching and mortality vary in space and time? What are those other stressors? What sort of adaptation, acclimation is going on? What's the impact of disease and other factors? And one of the big things is that the thermally driven mortality has surpassed the local factors in many cases and is getting worse. The ocean acidification folks have been working toward a more holistic approach in understanding, and that means an increase in complexity and understanding the details of what's going on. And what are the mechanisms of resilience to ocean acidification? What are the biological and ecological responses that fit into that? And what they've generally found is that the early thoughts and concerns about ocean acidification were merited, but as we get into the details of species, of locations, of interactions with other stressors, it's a very complex problem. And we need to understand that if we really want to help managers out and provide them with the information that they can utilize. And finally, the multiple stressors. This is where things start to really get complicated, but is so important. Heterotrophy is important. Learning that, in fact, in some cases when there's warming, some degree of sediments can actually be good, but not too much. And how much is too much? All things need to be done in moderation. There's an importance of natural gradients, of refugia, and of adaptive responses of various sorts. But what are these links between physiology and the microbiome? How does that microbiome play a role in responses? Where are those refugia? What are the factors that lead to it? And what species, what populations, or even what genotypes are the ones that are resistant? Because warming doesn't happen in isolation. And we really need to be looking at these multi-stressor responses because that's what the corals are seeing. So just to sum it up, the 500-pound gorilla of climate change has really won. It's surpassed local variability in most cases, and it's effectively killing corals at a very widespread area. No individual stressor is an island, they all interact, and we don't have the choice of voting to Brexit. We can't leave the Earth, we have to fix it. Thank you.
Thank you, Mark. That was a great start to this. So we're going to go ahead and go through the 3 talks and then have the panel. So our next one that's up is diseases and coral microbiomes.
Thanks very much. You guys rock that you're still here. It's been a really long week. It's been some pretty incredible news, not all of it good. I'm proud of you that you're all still standing, or at least sitting here. So this is Greta Abbe. I'm Drew Harvell, and we're here to report on the coral disease and coral microbiome, not the invasive species. That was, as it turns out, not part of our job. How do I advance? Green button. Green button. Okay, so first for microbiome, we were asked to do a very simple summary. So really it comes down to the questions that were asked is who is there and what are they doing? So the big issue, the big advances in this session, I think, is what are they doing and how does it vary with the environment? So what we were seeing is a lot of talks that are moving beyond the sequencing of the bacteria components as part of the coral microbiome and into the function of the actual transcriptomics of how their expression is changing with environment and how the different components are changing through time. And it actually, I think you'd agree that these sessions really completely interdigitated in the sense that many of the new innovations in untangling the biological interactions through the microbiome were actually measured as transitions often into disease and providing sort of a way of getting at some of these polymicrobial interactions. My key point here about 2 exciting integration of microbiome and disease transition, that doesn't really cut it. Disease is an amazing sentinel event in our corals, and it's an incredibly fascinating interaction between an invertebrate and its microbiome and its infectious agents. And to be able to actually be getting at the biology of how these transitions are made was pretty exciting. And I'm going to give you a couple of examples. And then again, in this session, again, it was 4 full days almost of seeing this unusual impact as sentinels for change. So let's just take a couple look at these. Oh, and then this came up yesterday. Corals, microbial sentinels, the coral microbiome will be key to future reef health. This was a paper by Tracy Ainsworth and Ruth Gates that was in Science. So here's one example. This was a paper by Joe Pollock, and I really loved this. I think is to just give you a sense of the spread of these talks and the scope of our advances in being able to actually handle these kinds of questions at this scale from coral microbiome. The first thing this group did is they basically picked apart the organism, so they looked separately at the coral microbiome for the mucus layer, the polyp itself, and the skeleton. And oh, wasn't it a surprise? It's really different. So we have to pull apart the constituents of our organism to understand the microbiome and how it, not only the constituents varying within those compartments, but their functions. And then, again, they looked at a range of coral genera, and not too surprisingly, we're finding widespread differences of coral I'm going too fast, microbiome composition across different species. And the surprising result here was that the life history mattered, that the coral biomes were tending to cluster a little bit with the brooders versus broadcasters. And then, of course, latitudinal diversity hypothesis is supported with changes. So this just gives you, this is impossible to kind of summarize this for all of these. The other one I chose was a paper by David Bourne and his group because it kind of highlights the microbial functions informing coral disease drivers. And, they picked one of the oldest diseases in the book, right? Black band disease, that's the one that's been described since Antonius for decades and decades now. And we're still, new innovations are coming out in that invertebrate microbial interaction. What David and his group did is they started with the black band cyanobacteria in the kind of the upper left of that, found out that it was critical what the level of photosynthate this pathogen is producing to kind of drive this whole cycle of polymicrobial interactions. How cool is this, right? It's not just this cyanobacteria, but there's a whole bunch of other heterotrophs that are piling on and contributing to eating up at the host, contributing to producing an anoxic environment, and then sulfate-reducing bacteria, and then coral tissue degradation. And it's, kind of a vicious cycle. But pretty exciting in terms of the science that we're unraveling some of these interactions. And then I just want to highlight one other study. This was one by Betty Willis and her group. And again, the value of now we're reaching the point where we've really got time, 13 years of monitoring. And this figure looks pretty complicated, and I'm not going to go through it, but you can see that these are some of the 5 most common diseases on the Great Barrier Reef. And they're actually working out what are the thresholds for a baseline when we're not worried about disease, as opposed to how do we actually investigate an outbreak. And I think I'm going to just keep moving on that. So to kind of summarize this, there's been largely 4 coral disease themes: detecting and monitoring disease, identification of pathogen, links with environment, and controlling disease. And I'm not going to tell you the star papers in those because I'm running out of time. So I think the really main point here is just the exciting integration of microbiome and using that to understand disease transitions. And I can tell I'm out of time.
Yes, you are. Great, thank you. Okay, another great summary. And so now we'll bring up the watersheds and pollution and sedimentation.
Thanks very much. I fully agree climate change is a 500-pound gorilla here in America, but water quality is still a 100-pound chimpanzee that can't be ignored, and we do need a full zoo in our ark. So Celia and I tried to go through this idea of summarizing over 200 tasks, 200 talks in this thing. It's impossible. But water quality is a key issue for at least 25% of coral reefs around the world. That's a probably conservative estimate, and the number is increasing because coastal pressures are increasing. But there's good news. I mean, although many of these pressures are local, solutions are local, so people have ownership and can actually make a change. And the even better news is many of the underlying mechanisms are global, and there's universal principles driving these things. And I think that came out at the symposium a lot, that what we found in Okinawa is also happening on the Great Barrier Reef in many of the small Pacific nations. So there is lots of scope to really start getting out of our silos and learning from each other much more than we've done in the past. I won't go through even a fraction of the talks that have happened. Just a few eclectic examples are, we have toppled dogmata at this conference. For example, there was a beautiful talk by Gordon and all showing how grazers are very strongly affected by sedimentation. In the past, we had this old debate about top-down versus bottom-up. Forget about it. Water quality is affecting the grazers as much as their food. We had a lot of new knowledge and tools, boys' toys, models, monitoring tools, and so on presented that will allow us to collect more data more efficiently and synthesize those data effectively. We learned a lot about new emergent pollutants, such as sunscreens, microplastics, coal dust, harmful algae blooms. All of those things are affecting corals and therefore the ecosystem they're building. And we learned a lot about mechanisms of physiology and molecular tools, how to better monitor, how to, for example, the imbalance between nitrogen and phosphorus affect the thermal tolerance of corals. So I think we're in a much better position now to implement actions. Or other examples. We really disentangled very complex pathways, like Celia's work on the virus tumors in turtles, tracking it down and really creating the links to the nitrogen pollution through groundwater injection wells that is taken up by the algae, and that leads to the tumors. We also heard a lot about management solutions, and there's really a greater acceptance now about the value of catchment to reef, ridge to reef, ecosystem-based management solutions. But the one big problem is really, despite many of the problems being not subtle at all, like this turtle, and I could have shown many other pictures, very often the still incomplete knowledge is used as an excuse to not act, or the willingness for some other reasons is not yet there. And I think that is really where scientists and managers are having a great task ahead to really make a difference. Also, water quality is a multi-stressor issue, and we as scientists quite often get bogged down in this huge complexity. For example, sediment discharge doesn't just cause smothering of corals, it also cuts down light, it changes the trophic levels between phototrophs and heterotrophs, and all these things, depending on our perception and our hobby horses as scientists, we're focusing on one aspect or the other and come up with different answers. We only found out very recently that the nature of sedimentation varies enormously the effects on the organisms. So there's a lot to be learned, and the turtle example shows how important it is to add science to come to management solutions. But in many cases, solutions do exist, we just got to do it. And all those pressures and solutions are local, as I said. Many underpinning mechanisms are universal. And because the problems are so local, I was a little bit scared at this conference how many silos there are. We are not learning enough from each other, and we are not getting out of our local perspective, seeing that the same issues are happening all over the place. So I think my take-home message for the ICRS is, and maybe for some conservation organizations also on, let's get the people together to learn from each other, to create critical mass, to share knowledge, to mentor local champions in all these places where there is super monitoring electronics don't exist, and really therefore empower local communities to create the local solutions that do exist. And maybe you also need something like the guest best management practices, which has been very effective in the ocean acidification research to empower people to do the science better and to have global and universal data reporting standards to get the whole science of water quality onto a new level. Thank you.
Now is where we can use some audience participation, and so please come up to the mics and address questions to our panel. We have a number of people who didn't get a chance to speak, but they're also up here to help address some of these topics.
I will act proactively, go forward, because I have to leave in 15 minutes. I just want to give a comment that will be in concert with what Katrina said. There was in the first presentation that Mark gave a very nice picture of, surviving corals, right? The 5-pound gorilla, it will be very dangerous to say, and I, by the way, have heard in some presentations that the management is unnecessary practically because everything is killed by the global stuff. It's not true, because those guys, they will die of the local impact. Those few survivors that actually can act as a pool that will propagate the new succession will be just simply gone with the local. And that is especially attributed to the overpopulated areas. And we have a lot of overpopulated areas, both in developed and undeveloped countries. And by the way, developed countries are as bad in overpopulated areas from the standpoint of pollution as undeveloped, probably even worse, because very often you can't see this pollution because it's mostly dissolved matter, but it's as bad. So be careful about 500 pounds gorilla. And by the way, also, scientists, please don't use feet, pounds, and everything in your, you're a scientist, okay? So.
So just a brief response to that, since that was in there. I won't comment on the feet and pounds. A 500-kilo gorilla. I don't know how much does a gorilla weigh? Anyway, the important thing is that when you're dealing with milder thermal stress event, good management probably can make a difference. When you're dealing with a really big event, it probably overwhelms it. But even in that case, the recovery can very much depend on the management that's going on. So you're absolutely right. We have to make sure that we don't lose track of the fact that good management still leads to more effective survival of coral reefs, even under climate change.
Okay, so I always wonder how we can bring the issue of water quality front and center to the politicians. It's a constant fight in Florida, for example. It's just constantly. So there's a movie that is called Erin Brockovich, and there's a great scene where there's all these people polluting an aquifer, and Erin Brockovich gives a glass full of the most stinking, dirty water to the lawyers defending the polluting party, and she asked them to drink that water. So I'm proposing perhaps we should invite all politicians and decision makers to drink the polluted water of the rivers that is being dumped into the ocean. And perhaps when you give them that kind of water, they will wake up and take some action. Thank you.
It's really hard to hear, isn't it? It's clear.
How do you bring water quality to the attention front and center?
We face that particular issue quite clearly in Maui, where the county makes decisions on how to handle the wastewater, which is an element of water quality. Decisions were made quite early and driven solely by the cost of the injection well implementation. It's taken us 30 or 40 years to figure out the ultimate cost to the ecosystem that's tied to the use of injection wells. If you have images like the turtle image that was shown you can communicate quite quickly the need to have healthier ecosystems. And the linkage that's we've found to be important is helping people realize that the delivery of that injectate is actually underground. You can't see it, and that was the missing puzzle. There has to be communication at a level that people can understand. And then the consequences being so dire that no one would elect to have that as an outcome. And possibly a lawsuit will help. But ultimately, I think everyone would not choose that path if given a chance to go back. And I hope that that informs policy and decisions for wastewater management, as well as agriculture, because the agricultural impacts can have just as profound effects. It's loading the system with nitrogen. It doesn't really matter for this disease whether it is an agricultural source or a wastewater source. Thank you.
Any other comments to that?
Yeah, if I could just address that slightly. Historically, the issue of runoff has been hampered by the scale of management and the scale of governance on coral reefs, which in many places focuses on the ocean rather than on land. And the same is true for the issue of climate change. So the coral reef crisis is a crisis of governance. And everyone on this panel is a biologist. We're not the social scientists who study institutions and how they engage with each other. That to me is a huge research challenge. And if I could give you just one quick example, and that's the Great Barrier Reef. The Great Barrier Reef Marine Park was established in 1976. It was an amazingly visionary thing to do all that time ago. At that time, it was the world's largest marine park. And it was designed de novo by an act of parliament to be the institution in charge of an area of real estate the size of Italy. It's 345,000 square kilometers. And it worked really well initially. But now the scale of that governance has been outgrown by the scale of the problems impacting on it. So coastal development is outside the purview of the Great Barrier Reef Marine Park Authority. So formerly the buck stopped with them. They were in charge of everything within the boundaries of that huge area. But of course they have no ability to deal with externalities like coastal development, runoff, and of course climate change. So to me the biggest challenge in managing and governing the world's coral reefs is to deal with the governance structures, and we're only just starting on that.
Timing. So I want to say a big thank you. I know not everyone got to be real active in the discussion just because we don't really have a lot of time this afternoon, but everyone here stepped up to a somewhat last minute request for the preparation of the slides and all of their participation in the panel, and I just want to thank them all again very much. And we're going to go ahead and bring up the next group of panelists who are Les and Bob and Dave and Sarah and Ileana, I believe, talking about, oops, sorry, I went forward. I don't know how to go back. I guess that's Ileana, right? Are you up there next? Wait. Yep, you're next, yeah. Yeah, try to do 5 minutes. Thanks. Good.
Hello, everyone. It's a great pleasure to try to summarize the sessions on adaptation, genetics, and selections. We had a really constructive and really innovative set of talks over the last few days, and there's been a lot of work, so forgive me if I miss some important points because it's quite difficult to summarize all of it. So I think what we've addressed over the last few days is really how corals can survive a rapidly changing climate. And that in and of itself, that question has really moved from if corals, or is it even possible to, yes, probably, how can this happen? And there's 4 topics that were addressed over the last few days: acclimatization, de novo adaptations, selection on standing genetic diversity, as well as hybridization. So in the first topic, under the first topic, acclimatization, research has really made progress in showing that, yes, previous exposure to stress has some impact on how corals may survive a stress event. There's also been some results on that might perhaps be a more short-term response. We also, for the first time, really heard a lot of talks about the potential role of somatic mutations in the host. It's a real active research area where I think we're still working on some methods and not quite yet at the stage where we can say what that role is. And the third real important progress here has been a brand new application of epigenetics methods on the coral host. And we're, again, had some really interesting discussions and quite some contrasting results. So I think by the time the next ICRS comes around, we'll have some resolution of the role of methylation in helping to transmit information of previous stress events to the next season as well as to the next generation. There was an amazing set of talks on the role. What really became clear was that in all of the talks, the role of the host genotype was paramount, and the amount of genetic diversity that we have is really, really astounding. So in every talk really highlighted how we have a large standing genetic diversity in our coral populations, and that selection can act on that standing genetic variation to help increase some traits and alleles to rise to high frequency. So that was really interesting as well. In terms of de novo adaptation, so this is really, evolving new adaptations to these stressors. There's been some exciting work on symbiodinium. This is done in the lab, where this is experimental evolution, really trying to see if we can get some de novo mutations, and there was some exciting results on that as well. And finally, hybridization has come up as a potential for a shortcut to get adaptation to advance more rapidly. This can be both natural as well as experimental, where you can combine the background of 2 species which are genetically quite diverse into a hybrid and see if there's some increased performance there as well. So I just mentioned this experimental evolution experiment showing how there is a potential for de novo adaptations in symbiodinium. The other, excuse the short symbiont there, symbio. The other really exciting talk that I saw was the suggestion that perhaps under really high stress, temperature stress, symbiodinium may switch to sexual reproduction or have increased sexual reproduction. So I thought that was a really interesting prospect there as well. And then finally, that microRNAs may play a role in the crosstalk between the host and the symbiodinium, and this may happen on both sides. So some real advances in using genomics and proteomics as well as metabolomic methodological advances and applying it to corals. So from my point of view, the key questions for future research is really, we've now documented this huge amount of standing genetic diversity within our coral populations. However, just the fact that there is such large variation implies that there are trade-offs, and we haven't found them. there's just a few suggestions. So where are those trade-offs, and what do we have to do to find them? And I think there's a real need for developing better methods in phenotyping so that we can really quantitative phenotypes and relating them to fitness that will help us find those trade-offs. What are we buying when we have corals that are stress resistant to thermal stress? What are the trade-offs to that? We also have not really been able to, not yet at least, been able to convert this into a more population-based approach, and this is part of that is, for example, measuring a selection coefficient. Finally, as I mentioned before, the role of methylation has been really sort of, it's exciting new field of research where we still are not quite sure what the role of that methylation is in really transferring some information transgenerationally. The interactions between host and symbiont have also not been addressed at the same scale as we have now done. The resolution we have for the interactions between host and symbiont is still a mismatch in scale, where the symbiodinium is still sort of these big groups of organisms, whereas we have a resolution down to the genotype for the host. So I think there's some work to be done in that area. And finally, once we have all those measurements of adaptation potential for the host and the symbiont, we need to ask the question whether or not that matches and what consequences that has for evolving a holobiont or selecting for holobionts that are stress resistant. Okay, so I think we're about out of time, so I'm going to leave you just with the slide, what I thought were the key findings. And again, I thought this was a real explosion of finding research in this area from the last to this conference, so it was very encouraging to see.
Thanks very much, Ileana. We'll bring up Les now.
Nope. Green, yeah, got it. Okay, first some general observations, then I'm gonna go weird on you for a few minutes. Like what else is new? The thing that impressed me most about the sessions that I went to was that everybody was reaching for something bigger, for some larger synthesis. We saw a lot of synergy between traditional and scientific ways of knowing, between theory and practice, between science and political action. We dealt with two great challenges, one of which wasn't mentioned that much after Chuck's paper, which is, of course, global climate change, which we realize is pervasive and deep and inexorable, but also export extraction and the realization that coral reefs are tightly cycled and they're not kind to commercial exploitation that involves extraction. But mostly, what I want to mention for a few minutes is the new vision that I feel like is emerging out of the symposium, new perspectives. And I want to talk about those. Here's where I go weird. I was all worked up for some more chance, so. If we look Mauka, if we look inland, we see that there's sort of a global plateau of reef health that's been eroded under the influence of our careless regard for nature on all kinds of scales, great and small. And it's been eroded into a jagged landscape with peaks that are like the reefs out of a time that few of us have lived to remember, and valleys of degraded reefs that are trapped in a sort of basement baseline. The landscape is really varied, and what we learned this week is that we're starting to understand how this heterogeneity came to be. We heard about research that could enable us to elevate the valleys in the landscape, to boost and defend the health of compromised corals and their human dependence, and also to enhance connectivity among the mountaintops, better enabling all the reasonably intact bits to become kind of a seed source that can lift the bottom up. Since there may not be quite enough time, we've talked of our efforts to turbocharge adaptation through direct manipulations. On the flip side of that same idea, we've learned over and over again about the gift of variability, about how this ENSO event, however terrible, has unveiled a broad spectrum of really idiosyncratic responses to stress, macroscopic, histophysiological, genomic, and sociological, that we can seize upon as a Rosetta Stone to what's going on underneath, what's really governing adaptability of reef organisms. You'll hear more from others about the progress in the science of coral reef husbandry and restoration, but just know that our momentum is building, and with a little more nourishment, we can move a lot faster, and just maybe fast enough. We've heard really fascinating things about functional and applied genomics, the clinical pathology of corals, and the design of reef restoration efforts at multiple scales. Looking Mackay, I don't mean to advertise the hotel, it's the best Mackay slide I could find. Looking seaward, we've seen the connections between land and sea, between people and reef in all their dimensions, physiological, biological, philosophical, and spiritual, more clearly than ever before. There were a lot of presentations on the way that reef fisheries could be made better if we followed Chuck's advice and developed laws that fit the biology instead of trying to squeeze the biology into hopelessly naive and wrong-headed laws. For those of us who still engage in the barbaric rite of eating them, fishes are delicious, and that fact easily overshadows the importance of biological diversity. Nonetheless, we heard about this from a lot of people, including Nancy, about all that we don't know and the humbling things that we're discovering. Of course, those reefs that support all the edibles and the inedibles are crumbling before our eyes, but not everywhere and not always. Reefs maintain carbonate accretion on the one hand, structural breakdown and sediment export on the other, and there were a lot of papers addressing this soft rock push-me-pull-ya, showing that here too, the intellectual ore to be mined is instructive variability. Finally, looking Mauna Wakea from the peak that connects the earth and sky from Mauna Loa. We've advanced greatly in two key areas. First, we've deepened our ability to see the ways of change over time through imaging technologies, in situ sensor arrays, and remote sensing. We've seen our colleagues bend this new data fire hose to powerful computational tools that make it easy to see the linkages between society and sea, the veins and arteries of the Hupua'a. We've also seen that work at this local system level can matter, even in the face of the global climate storm. Anyway, the wealth of data on systemic change over time yields the second pot of gold. The ability to view coral reefs as complex systems, to plot their time evolution mathematically and in a holistic but scientifically tractable way. We've heard lots of our colleagues draw attention to stochasticity, to chaos, to embrace the fundamental principles of complex systems, and to savor the lessons of uncertainty. There isn't time to understand everything, but there might be just enough time to capture the manifold that describes where it's all coming from and where it could be headed. With this nuanced view in hand, it suddenly becomes possible to nudge reefs positively, intelligently, restoratively, with just the right mix of knowledge, courage, and caution. Bob? Oh, you've done anyway. Yeah. Okay. Okay.
That was very elegant. I think you should get a Pulitzer for that.
Not a Nobel Prize.
Right. Well, okay. I was asked to talk a little bit about the what I heard in terms of reproduction moving to recruitment, and I think I could kind of bundle it up into saying that it looks like from the simplicity that we had in the past, that now we're looking at what are the factors that drive larval availability of whether fish, corals, you name it, the propensity to settle, and the availability of nursery grounds. And some of the interesting things about larval availability that I heard was significantly improved oceanographic models, connectivity models, but now being much better verified with things like microsatellites and trying to actually literally figure out connectivity from a genetic sense, putting real kilometers on it. But just getting there, it may be half the fun, but it's not half the story. In fact, It's the propensity to settle that is often being disrupted by a lot of anthropogenic things. We heard about ocean acidification affecting larval behavior, sound significantly affecting how larval fish detect the places they're supposed to go or avoid their predators. And they can pick up the sound of big ships kilometers away, and it significantly affects their capacity to settle. What are they settling into? The composition of the benthos is obviously changing, and available nursery habitats, at least for a lot of organisms, can be summarized: algae are bad, corallines are good, and there's a lot of dynamics driving that. I don't have time to get into that. Thanks.
Thanks, Bob. And now we're going to bring up Dave Vaughn to talk about reef restoration.
Thank you very much. You know, Sarah and I really had a hard time trying to figure out what of all the good news that's taking place in reef restoration we can say in just a few minutes. So we decided to say it as quick as we can in just one picture. And this one picture is probably something of the one on the upper left and the upper right that most of you have in your PowerPoints or slide presentations to the public, explaining to them what is happening with the reefs today. A typical picture of something reef in the 70s, and then a picture in maybe early 2000 and where the staghorns are gone and the palmata's gone. But I'd have to tell you that there's good news with reef restoration. We're literally changing that arrow direction around. So if you look at those bottom two pictures, these are actual pictures before and after reef restoration in Belize. So what we hoped of years in past is as just a dream, or at least a Band-Aid, has really had some good news as success today. So what are the key findings and updates from it? There are actually a lot. The number one thing for you all to realize is that for many years, the restoration had a few people. The restoration session had a couple of what if we could do something. The sessions were packed, and they were full of good news of it working. So restoration is possible, it's working, and it's a great alternative of good news to start to tell people without having to say that we don't need to stopping those stressors that are ruining it. There are a lot more projects. Many of the projects, such as in the Florida and the Caribbean, were because those areas had problems early on. There's over 30 different restoration projects in Florida and the Caribbean alone. There's many in the Pacific and the Indian Ocean. And I think we're just at the beginning crossroads of what will be a new tool for the toolbox in the future. There's not only just more projects, there's new discoveries. What started off originally with just work with one branching species has now moved to the other reef-building species, as well as to the potential of sexual recruits being possible. Never thought that the dream of sexual reproduction, not just asexual fragmentation, would be a reality, but I'm here to tell you, as of this meeting, sexual reproduction is a real toolbox potential that will increase tremendously. I was real pleased to see the amount of paying attention to genetics that was taking place by all the partners working on it. They were very aware of keeping genetic diversity and very aware of looking for resiliency. And in addition, socially, almost all of them are including local communities, education by students, and volunteers or citizen scientists. More hard coral species are now available, including all of the reef-building corals, not just the branching corals, and the ability to produce them by the tens and the thousands. And a lot of other reef species are being looked at. So what are the key questions for future research? How can we even improve beyond just the getting now tens of thousands of recruits successfully from a spawn, but even millions in the future? How can we implement more locations? We really need to do this at scaling up. And as we Sarah and I said, not just from square meters, but square meters to hectares, and why not to oceans. We need to improve and work on genetic diversity and resiliency, better monitor our success in ecological succession, and still looked as an example of why we have to reduce those stressors that inhibit recovery. What are the main points that IRC members we would like you to take home? Some of the biggest ones is that we're using scientifically based processes, not just dabbling. We've gone beyond just small scale and now being able to have something that works. It's a tool for you. Restoration complements the conservation types of activities. The new technologies are working and improving rapidly. And probably the biggest take-home message, sexual reproduction is now feasible and improving. We now have a number of tools. Now it's our job to put those tools together. If we can now sexually reproduce strains, which we know one strain may be resistant to one factor, one to another, we may be able to make crosses, get high percentage of recruits to make it, test them for their future resiliency, and now produce thousands of them for outplanting. So those tools now make a system that we can all put together, and that's going to be the trick, putting it together. And if we've had this much progress in reef restoration since the last 4 years, think about what it may mean in 4 years from now. So much so that I think reef restoration people want to work closely and more often. And in fact, they're going to meet today at 6:30, so at room 307, because we can't wait even another 4 hours. Thank you.
Thank you, Dave. We're going to now let the panel run for about 10 minutes. So, questions?
Whoa, it was stunning.
I'll ask a question. So I think every one of you touched on genetic diversity and maintaining that. And we know that things are changing, that natural selection is going to happen, and we're also doing a lot of, acceleration of the natural selection through various processes like restoration. What are the risks of that, and how do we minimize those risks? What is the best tactic across all of you to do that properly?
So I think if I can start here. So I think what we really saw is that, which is really encouraging, is that much standing genetic diversity in response to stressors allows us to select for stress-resistant strains, both in the host and the symbiote and the fishes and so on. The real challenge is to not just go for the winners. I think the best, because I think the trade-offs are there. And Roberto gave a very powerful talk reminding us about those trade-offs. And but we don't yet know where they are. Can we wait? Probably not. So what we need to do, I think, to safeguard against selecting for the wrong thing, or finding out what those trade-offs are later, is we can select the winners in our active restoration strategies and so on. But I think it behooves us to also protect and select some of the losers, so that we just get the whole diversity, or encompass the whole diversity. I think that would be a fairly easy thing to do without having full knowledge of every minutia that might be going on.
If I can add to that as well, when reef restoration was just basically fragmentation or cloning of many of the same strains, it was always imperative that people look for more resilient species or strains, but always keep all of the genetic diversity included, because we don't know what traits in the future are going to be needed from something that looks like a loser today, but maybe a winner in some other facts. The best news is that now that we have the potential of really being able to produce corals with sexual reproduction, we can increase the genetic diversity by making those crosses and having more diversity in that.
Speaking of restoration, a question that I have some great advances and things that have been going on. I'm wondering, with the size of the events that are going on and the extent of damage, what are some of the hopes you have in terms of being able to really scale up restoration efforts to be effective on a large area?
Right now, we have the techniques to go large scale. We've demonstrated we can do it. The biggest limitation, as I see it, is money. We need, which also brings another issue, we can access a funding pie that is different from the funding pie for conservation. So we are not taking money away from the conservation funding pie. We can find our own funding, but we need to target the big funders. And so that's the major problem to scale up. It's not a technical problem, it's a problem of just accessing the money.
On more of the aquaculture type of production scale, I think scaling is not the fear that everybody should have. We've seen the scale being able to go from people producing a few thousand to producing tens of thousands. We now see accumulations of 100,000 corals being planted in an area. And I've talked to 2 or 3 other people who have the same goals of being able to plant 1 million corals in the next few years. What we need is more locations. We need a lot of training and outreach to people to show them how to do it the right way.
I couldn't agree more about the good news about reef restoration. However, also the restored reefs will face, I think, the gorilla and the chimpanzee. So my proposition is that we need refuges, and probably we could also create refuges. I heard this morning that it's not always possible to have deep water in the area where the coral reefs are, but if the deep water doesn't come to the coral, we could probably bring the corals to deep water. So my proposition is to create refuges in open ocean areas, in deeper water, far away from the chimpanzee, and also protected against the gorilla. And I would like to hear your perception on this idea.
Okay, it's an interesting idea. What you are proposing, if I understood correctly, is that as we do coral reef restoration, that we target deeper areas. Is that what you were saying, right? Because we have to prepare the corals for this future that is coming. And that's an interesting idea, but what we've seen is that you're going to have a limitation at what depth the corals are going to get enough light for the zooxanthella. So the other thing that it's also being proposed is this assisted migration, where you start moving corals to higher latitudes, but also has another sort of problems. So one of the things we say in restoration is that we understand there's this overarching problem of global climate change. And what we need to do is work together with conservation. So we are in this race against time. So if humanity as a whole decides to stop burning fossil fuels, we'll have a chance of coral reefs surviving. And if they don't, if we don't make this decision, it's game over anyway, whether it's conservation or restoration, it's going to be game over. So those are the issues we're dealing with.
Okay, I just wanted to mention that I didn't mean to put the corals on the seafloor, but make moorings on the seafloor and make suspended reefs, let's say at 15, 20 meters depth, in that way. So they would have enough light.
Well, thank you very much to this group of people. Again, they did a wonderful job, I think, summarizing very broad topics, and we really appreciate it. So we're going to bring up the final group of participants and reporters, and we have Mark Hickson, Alan Freelander, Josh Sinner, Jack Kittinger, let's see, Jim Bonzak, and Allison Green coming up. And Mark and Alan are going to lead us off here. Again, we're going to try and do 5-minute talks, 3 in a row, and then have the panel. And this will be the the final group of reporting. Just this big green button.
Got it. Thank you, Val. And aloha to all visitors to Hawaii. Besides the corals themselves, fishes are obviously the most conspicuous inhabitants of coral reefs. And they are, in fact, the most diverse assemblage of vertebrates on the planet and an extremely important source of protein for nations living adjacent to reefs. When Al and I were given the task of summarizing all the hundreds of talks and posters having to do with fish and fisheries, we knew we were on a fool's errand. But being a good fool, at least on my part, we decided to try to take it up. We asked session chairs and various experts who were attending all these hundreds of papers to give us some general points for taking home. These are not particularly new points to practitioners, but we hope they're especially useful to the public and especially useful to policymakers. So the first point was that fish need healthy reefs. This has been pretty obvious for a long time. Reefs provide a place of shelter and food for reef fishes, but we're increasingly finding that even though dead reefs provide shelter and some sources of food, it's really the healthy reefs that provide the appropriate settlement cues for larval fish to find a place to live on the reef. That's a relatively new finding. Reciprocally, the reefs themselves need healthy fish populations, and we cannot overemphasize the importance of herbivores, not only fishes, but also sea urchins and other invertebrate herbivores, in ensuring that even a reef that's hit hard by bleaching, the dead surfaces are kept clean of macroalgae so that corals have a possibility of recolonizing and growing. Fishes are also extremely important in terms of nutrient transfer, not only in their feces, but also their bodies moving in and off various habitats surrounding reefs, and as a trap for plankton, for example. Predatory fishes affect lower trophic levels. That's becoming increasingly apparent. Predators in general among reef fishes are a very important source of what's called density-dependent mortality in their fish prey. That is a type of mortality that tends to regulate prey population dynamics. And that is extremely important if we want to conserve particular populations or manage them effectively as fisheries. Predators can cause cascades in trophic levels where abundance of one predator can reduce the abundance of the next trophic level below, which allows a release from the trophic level below that. And those dynamics just demonstrate that everything on reefs are connected. And in fact, I've always believed that's the greatest take-home message from coral reefs. It's all connected. And of course, top predators are extremely overfished. Not only the sharks, but also large groupers and all the larger fishery species, cause for great concern. We're increasingly finding that fish move. Don't have particularly small home ranges, but many do return home one way or another. The larval dispersal sessions show us that the good news is that larval dispersal tends to be a combination of both local retention or self-recruitment, or larvae spawned in a particular location do come back to their home reef or home general area, but there's also then long distance connectivity to other areas. The good news there is that spatial management can work because that system itself that's protected can be self-replenishing, but then also seed areas that are less protected. And then, of course, there's spawning migrations at all scales on reefs with fishes, daily migrations just a few meters, but also these very long-term, hundreds of kilometer migrations by grouper and snapper. And spawning aggregation sites are getting hammered by fishing and are an easy target for protection. Not only just where the spawning aggregation is, but also the surrounding areas, so the fish aren't all caught when they're on their way to that migration, to that site or leaving it. Reef fishes often depend on increasingly threatened non-reef habitats. Again, that connection. Mangrove and seagrass nurseries are extremely important for many reef fish species. We can't just protect reef fishes by protecting reefs alone. Big, old, fat, fertile female fish, affectionately called boffs by the people who study them, have been shown increasingly to be the most important spawners for reef fishes. And the way we protect these big fish is one of two ways. Either we allow a certain portion of the population to be completely unexploited in marine protected areas, or we somehow impose slot limits where only medium-sized fish are taken for food. Getting more into fisheries now, we're increasingly seeing that reef fisheries cannot sustain intensive exportation, either by intensive commercial fishing, the live reef fish trade, or what have you. The production is such that it can support the local population, as it has done for centuries and centuries. So movements such as by Palau to keep that fish production within the nation is extremely important. Overfishing of coral reefs is widespread, very well documented, and larger species, the larger food fishes, are especially threatened: large grouper, large wrasse, large parrotfish. So for solutions to these issues, holistic, often called ecosystem-based, community-based management shows great promise. There's examples starting to sprout up all around the globe here and there. And it's especially effective when traditional ecological knowledge that has been known for centuries by elder fishermen is blended with cutting-edge Western science. And we'll be hearing more about that later with the outcome of the Leaders Summit. Novel data-poor management approaches are required for multi-species, multi-gear, poorly monitored fisheries, especially in the context of precautionary approaches. Adaptive management is absolutely essential. Even the old ways that have worked for centuries may not work in the future, because the future will not be like the past. And then finally, in addition to these topics, some things that are coming up in the reef fish world are these low-cost, high-volume, high-resolution genetic approaches, which are going to really give us a good handle on larval dispersal and connectivity, as well as phylogeography and evolution. And mesophotic reef ecology is starting to come to its own. Thank you.
Thank you, Mark, and that leads nicely into Jim Bonzak's going to continue on with some management.
Aloha. So we have kind of a eclectic set of, well, the big green button, topics to cover. First of all is the remote sensing and emerging tools. And this is one of the real highlights of this meeting. There's all kinds of technology being applied, from satellites to underwater gliders. We can do any kind of wavelength now in terms of monitoring surface things. We're learning about currents, turbidity, color, even bioluminescence. So that's really a success story. Tagging has improved greatly, both acoustic and satellite tags. We actually have tags now you can put on a fish. They don't even have to be caught again because they swim by a sensing buoy and it'll dump all the data for you on a sensing buoy. So there's amazing things there. Autonomous vehicles, hexacopters, gliders, AUVs are all becoming more inexpensive and more reliable. Optical technology has improved greatly. GoPro cameras, underwater stereo video. So there's quite a bit there. But also the innovations, there's a lot of talks about citizen science, how we can multiply our ability, assuming we get citizens to do the science we need. But they're certainly dedicated and helpful to us if they're properly cultured. And I just put eDNA. There's DNA, everything is being done, but the eDNA was kind of more interesting because you can just take a water sample and try to figure out what the biodiversity is. So I think that's a major advancement, but it's across the board. We focused on MPAs, a lot of talks. I tried to do a little enumeration, I lost track. It's sometimes not even clear in the title, but a lot of focus on the scale, location, what works, what doesn't work, how can we design these better, are they effective or not? Clearly they're effective for fish. The effectiveness to coral is not so clear, and that's may take decades to solve that one, but certainly that's one of the issues. The impacts on goods and services, not only just improving the science, education, cultural resources, and the non-extractive activities. They also provide one of our tools for monitoring. That's our control, the area that's not being manipulated by extraction. So it's important for understanding of how systems work. Baselines aren't there anymore. And a lot of talk about governance, compliance, and effectiveness of marine protected areas. But the important point there in the little figure is that fish are not the same. They have many different home range sizes, behaviors, and some of the, to protect the larger fish, probably going to need very large reserves indeed, and a lot of discussion how much area should be included. Fishery management, just follow up a little bit. This is probably one of our major research obstacles, and that is that fishery management is really based on four things: allocating who gets to catch the fish, determining allowable fishing gear, regulating the size and amount of fish can be taken as in bag limit or size limit, and limit effort mortality to be sustainable in the long term. That's what fishery science does. You don't see anything about ecosystems there, habitat protection, but that's what traditional fishery science does. The point I want to make here is using marine reserves as complement. It's an ecosystem-based habitat approach that eliminates bycatch, targeted fishing, habitat damage in the areas that's protected, and it complements nicely the traditional fishing tools. One of the problems we have, though, is not being highly adopted by fishery management. It's been a real task, and it's one we'll face, but it's one we need to look at in the future. And I want to emphasize both are necessary. You still need traditional fishery management, but we also need new ecosystem approaches to management. Take-home points for ICRS people and the public is this, that MPAs provide biodiversity protection. They can help with fishery management. They may provide some protection for climate change by maintaining healthy areas, less impacted, and certainly for tourism, management, recreation, non-extractive activities. There's a lot. We have major universities that can't take their student out and see a healthy reef, for example, in a lot of places. So those are important. And finally, get into the idea of the theme of the science to policy, policy to action. And this is kind of a little cartoon describing our role in science. The idea is we monitor the human dimension, what people do, and their reef resources provide advice to managers who hopefully develop policy and regulations. But there's some bigger issues there, and I want to emphasize the importance of communication, that is something we need to work in, one of our challenges. We do a fair job maybe of communicating with the public, not so good maybe. They do a great job of communicating with us. I deal with fishermen and MPAs, and their message is very clear. They don't want any restriction of access. They want no restrictions on their activities or changing their behavior. We do not need MPAs. I'm not the problem. Someone else is. And all solutions must be painless. So it's pretty good what I hear, but we are not doing a good job. They could be important allies if they have better understanding of some of the issues, I think. But the biggest one is our relationship with science and management, and that is working with managers. And this was nicely summed up today in a talk by Jane Hawkridge this morning on our research challenges, is trying to figure out what the management targets are. I do this all the time. What do you want? How much change is tolerable? What kind of precision do you need in our estimates? How much risk are you willing to accept? What do you want to know? And how much science can you support? So trying to live up to Les Kaufman's always elegant oratory, I tried to find a profound, fundamental, philosophical, existential quote to close with, and I think I found it. I want to give due credit to the Spice Girls. So tell me what you want. picture disappeared. What do you really, really want? I'll tell you what I want, what I really, really want. But that's one of our biggest obstacles to research, so thank you.
Okay, thank you, Jim. And finally, we're going to have our final report by Josh Sinner.
That was a hard act to follow, so I brought some help, or hopefully we won't do a dance. No, as social scientists that study cooperative governance, we decided to practice what we preach, and we're going to do this one together. So one of the things that I've really noticed about this talk in terms of this conference in terms of human dimensions, there's been an increasing recognition that reef conservation is primarily about people. That you manage fish, you manage people, you don't manage fish. And I think Terry Hughes put it very eloquently with the notion that the coral reef crisis is really a crisis of governance. So social scientists often gets the rap of being sort of the quote-unquote soft sciences, but even Nobel Prize winning physicists recognize that they actually have it easy, and that people are very difficult, and if you've ever met one, they lie and they cheat, and they're terribly difficult as subjects. You guys are very lucky with corals and fish. So I was asked to say what were the key findings and what were some of the updates to what we think. And I think there's been a real, so this is my 4th ICRS, and there's been a real sort of evolution in the complexity of how we think about human-environment interactions. And I would say that when I first started coming, the majority of the problems were framed largely like this, that there's bad things happening to reefs, and that was bad. I would say that started moving towards a notion that, there's a lot of, that people are dependent upon it. And this came with the Millennium Ecosystem Assessment Framework, which really talked about the different ways in which people benefit from that. But I think one of the great things that's come out of this particular ICRS was a further recognition of the real complexity of it. And it moves from what social scientists would call the proximate drivers, i.e. that fishing is the problem, to understanding what we call the ultimate drivers, or the distal drivers, i.e. that things like markets and poverty and a whole range of social and institutional and governance structures are shaping the ways that we interact with it. So it's not just that overfishing is a problem, well of course it is, but what's causing the overfishing. And I think we've seen an expansion in the ways that we do that. And just a very quick example, there was a whole session on sort of supply chains, and I think that's really critical that we're not just thinking about these in really simplistic terms. And this morning in my talk, I mentioned the sort of increasing sophistication moving away from what social scientists would call a Malthusian view, in that the number of people are the problem, and therefore the solution is to create situations which we lock people out. We're taking a much more holistic view and understanding the broad array of drivers that are causing people to behave in certain ways. And the reason that's important is because it opens up different solution spaces, right? If you understand that markets are the problem, you can tinker with supply chains and end up with different solutions. And so with that, I think I'll actually turn it over to Jack.
Thanks, Josh. I had the opportunity to see the New York Times columnist Thomas Friedman give a talk recently, and he was asked, what are the 3 primary forces shaping the world today? And he said, markets, Moore's Law, and Mother Nature. And it occurred to me that those were a lot of the themes surrounding a lot of the social science research and human dimensions research that were shared at the conference. Markets, because our globalized economy is bringing demand to reefs in new and very destructive ways, but also that there's solution sets in those things. Moore's Law, because the technological advancement of the way that we interact with each other and with the environment is just growing at an incredible rate. And Mother Nature, because we have profoundly changed the chemistry of the planet, and the consequences of that have become unbelievably undeniable. And I asked a friend of mine when we came out of some of the climate change sessions what she thought about some of the talks she had seen. She said, well, it's a bad year for good news. And I thought that was pretty accurate coming out of Terry Hughes' talk about the scale of bleaching that is going on. But through the week, we've seen an incredible focus on solutions. And communities are just doing incredible things. And they're doing incredible things on all these social factors, that are really the key of how people come together collectively to take action. And we're getting much better at measuring things on the social side. We're getting better at quantifying how people benefit from reefs, how they impact them, what is the capacity to change human behavior. Ultimately, that is the key. And in essence, that's kind of the key here is the science of successful interventions. You know, how do we take the world as it is and transform it to the world as it could be? How do we intervene? And I hope in ICRS 2020 that we have an increased dedication towards testing these solutions and sharing what works and why. And because there's kind of a selection bias against talks about failures, I would propose that we also have a fail fest session where we talk about what doesn't work. You know, that might be good for the evening session where a little liquid courage could go a long way. And then the last thing I want to say is that I think we're going to do ourselves a disservice if we continue to make the coral reef crisis about reefs. It is not about reefs. It is about us. It is about the people that depend on reefs. It's about the father whose new dive tour operation in Indonesia is threatened because the entire ecosystem that he brings people out to see is gone. It's about the woman who runs her cooperative. It's about the family displaced from their atoll. Those are the stories that people will respond to, because your average person, doesn't matter if they're in Nashville or Nairobi, does not care about reefs. They care about people. And that's what we need to make this about. Thank you.
Thank you. That was an excellent summary. So let's go ahead and have some questions for our final panel, and then we'll move on with the program.
Yes, my name is Anders Paulsen. I'm Artistic Director for Coral Guardians, which is a project within Stockholm Resilience Center. And I would like to highlight the importance of connecting with musicians and artists to emotionally engage communities to environmental stewardship. Emotions change behavior more than intellectual information. And it's not just about outreach, it's also about inreach. Music is not just a luxury for the ears or just entertainment, but it's a valid tool in the toolbox for bringing people together and for communicating the urgency of better stewardship. As a professional musician, I worked in 27 countries, and I meet musicians everywhere who would like to contribute to environmental stewardship and awareness with their art. So there's so much goodwill to be harnessed. And finally, respect for indigenous culture, celebrate the beauty of the earth, because beauty is a necessary ingredient for human well-being. And also spiritual engagement to care for the reef and honor it like the sanctuary that it is. Thank you.
Okay, yeah, the only thing I'd just mention on that is that, I think you raised an important point that it's not just information that changes. Human behavior is governed by a range of things. Social norms is a key one, but it's not just information, right? And I think that's the key point that you were saying. And so we have to do more than just provide information, because that's often, so that's what social scientists would call the information deficit model. And if we just tell people something, they'll change your behavior. But anyone know anybody that smokes? Right? Does that person know that it's bad for you? Right? I mean, so people know things and they continue to act in certain ways. And so, social scientists have studied how to change human behavior for a long time. And so it does deal with connecting, sure, with emotions, with art, but also with social norms. And that's one of the key ones.
Can I respond? One other thing is that one of the strongest things to motivate people is their self-interest. And I go to Aldo Leopold pointing out that you have obligation, responsibility, and self-sacrifice are necessary ethics to maintain our environment, because we are part of the environment, we are part of the ecosystem, and so the things you enjoy doing now, you'd like to do that in the future, you should protect it. If it's valuable, protect it. So I just want to add that ethic aspect in there. Yeah.
I know we're standing between you and beer, so.
I'm going to ask one thing just to bring it back to the science, and that is, not that that's not science, I think that art is a very important part of that, but, you know, we've heard, and this is to all the speakers, I think, you know, what is the role of ISRS in the next 4 years in taking, let's start with what Ruth was talking about in terms of taking some of these ideas, building real networks to speed up the process of understanding some of the tools that we have and applying those tools and actually discovering what the failures are, what the successes are. What do you guys feel is the best thing ISRS can do right now to form these networks or just really speed up the process that we as scientists need to do to form these communities to get this work going quickly? If you got any ideas, it'd be great to put them out there.
I'll dive in there. We need to bring coral reefs to the people in a way that is compelling and touches their everyday lives, just as Jack reviewed. And I believe a very good starting point was Bob and others' idea to have this leaders summit at this meeting to tie the scientists and the technical expertise with the policymakers, because the time for study really is past. We know enough that we know what to do. The crisis in governance must be addressed, and it's going to be addressed with not only educating the governance, but also bringing the people up from the ground up to demand change. Thanks.
Sure. I think there are a lot of communities working throughout the region. There's been a renaissance of community-based management in Hawaii and throughout the Pacific, where communities are taking charge of their resources from the bottom up, recognizing the knowledge that existed within the community, taking advantage of that, and as far as you know, recognizing shifting baselines and having to, you know, they're taking the initiative to establish management practices that they used in the past that are revitalizing, that worked in the past, but they're contemporizing those. And I think that that's really valuable, but what's lacking in a lot of places is the top-down support for these bottom-up approaches. They can't go it alone, they need some facilitation. But there's a lot of knowledge that's locked up within these communities that far surpasses any of the knowledge we can ever acquire scientifically, and working together with scientists that helps to elevate that knowledge and then empowers them, but they really need the top-down support, which I think is lacking in a lot of places.
If I could add also, I think what is often missing is just the right information in the right way. The fish movement slide that we saw just a few minutes ago has really opened the door to much better design of marine managed areas. And it's not rocket science. It's like, if you want to protect a fish, your area's got to be bigger than how far it moves. But people didn't have the information in their hand to make the decision for the fish that they cared about. And so, for example, we went and worked with people in Ponape, and we showed the fish movement stuff, and they said, we already know this. The fishermen said, we already know this. I said, well, have you ever thought about
what that means for how big your areas were, and this penny just dropped. And then suddenly they did this analysis and they've made bigger areas because they get it, it's not rocket science, it was just the right information in a very simple tool that they understood. And I've seen communities in many places around the world make much better decisions on their designing their areas just by that simple communication tool for something they already knew.
And Austin, I might just say, yeah, I mean, there are examples of these sort of integrated conservation and development projects, and Georgina Gurney presented one on Monday evening, and it looked at sort of the impacts of both conservation and development on community well-being over time, and it was a great example of how that, you know, that sort of integrated conservation development project can improve people's well-beings.
This one especially to the social scientists, but anybody in the panel. I think we should modify a little bit the message about why we should conserve and restore coral reefs, because the message has been so far because it's for our children and our children's children, future generations, and so forth. But for what I see, I don't think our species really cares about our children, because if we did, we wouldn't be in the trouble we are in right now. I'm pretty sure everybody here in this room, they really care about their children and their descendants. That's why we do the job we do. But there's a world out there who doesn't care. I mean, they're only focused on, their attention span is 140 characters, and that's what it is. So I think the message should be that we should conserve and restore coral reefs because it matters to us, because in our lifetime, we're going to suffer the consequences. It's in our lifetime we're going to see the changes, and we don't want to get there. And so how could we rehash this message to just make it relevant to our lifetime? I've seen changes in 10 years, in 20 years, and biologically I'm supposed to live many more years still. So I'm really concerned what I'm going to see 20 years from now and how that is going to affect me. And I think if we rehash that message that way, perhaps we'll have more reaction worldwide. Thank you.
I was going to address Joni's challenge, asking about, you know, what ICRS can do. And this meeting has been very good, I think, in this synthesis session is one example of how we can share information. And I'm wondering if meeting every 4 years is too long a time. And I'll give you an example. I think the genetics session and genomics sessions have really shown how adaptation can be extremely local. So on very, very small scales. Now, what does this mean for reef restoration? It means that if you're moving your genotypes from the fore reef into your nursery and then back out on the wrong type of reef, it's not going to do huge damage. It just means that that genotype is going to die because it's in the wrong environment. I think this is something we've learned quite clearly this time around. But you know, now you ask me, well, what's the right environment? And I think we don't have the measurements yet to measure this on the right scale. So we need to make some pretty quick, you know, improvements in this. And 4 years may just be a pretty long time to share that. So I'm wondering if we need to go to more frequent meetings.
One issue that's helped with me in my talks and dealings with the public about coral reefs is just to bring it home one way or another. And I have actually found that people with children do care about the future of reefs just because of their intrinsic beauty. Fortunately, my daughter was very kind to give birth last week to my grandchild, and that's just resolved. That's just resolved my own path. But ways to bring reefs and people together are to make, you know, direct lines. I mean, one thing that seems to resonate with audiences when I speak are all the novel anti-tumor drugs that are being derived from coral reef organisms, because almost everyone's been touched by cancer directly or indirectly. And the thought of having maybe not lost that loved one if that particular reef hadn't died does resonate with people. So there's always ways to bring it home. And ultimately, again, it's about people. Thank you.
All right, I just have a quick suggestion since we're at what ICRS could do better, and I'm definitely a big supporter of this happening more often than just every 4 years. And I think these headlines that we saw during the summary sessions now, they should be on big signs in the lunch area, in designated areas. That lunch with a mentor thing was a good idea, but I felt like the lunchtime is the best time to promote even like some sort of random collaboration. As you guys rightfully said, we have to work more together, but I didn't feel like walking around these tables and going like, oh, do you work on ocean acidification? Do you work on ocean? Like I wouldn't know who works on what. So not that people have to sit at these designated areas, but maybe that would be helpful to just put up big signs so people can sit in an area surrounded by other scientists, many other scientists doing the same thing, and that might actually promote more collaboration. Thank you.
If I could say one last comment about the previous question about what ISRS can do. And to my thinking, I think there's a big opportunity space for the incredible scientific ability, the knowledge base that exists in this room, because this meeting is dominated by scientists. There is a lot of managers, practitioners, and increasingly political leaders that are attending these things, and that's a huge sign of optimism that we should also be proud of. But there is a huge opportunity space, I think, to link the practitioner community more directly with the incredible work that all of you folks are doing. And that's something that I think this society could really prioritize over the next 4 years. And I hope that in 2020, when we're back together, that there's a lot more stronger links in that regard.
Yeah, I agree.
Well, it's good to have the practitioners up here. It's an appropriate session.
Exhibit A and B.
Yeah. Thanks. Thank you.
So just a couple last things to do, and then we're out of here. So I first really want to thank Val and Joni for putting together what I think was a really valuable session, and just getting people again to talk about that it doesn't end today in about 10 minutes, but rather this is an ongoing process, and we really need to figure out where we're going next to make it work. I just want to let you know there's been a couple of very important outcomes that were achieved this week. One of them was after some alcohol last night, where a group came up to me and said that they were so inspired by the work that they saw here that they're looking to put out a challenge and that they'll be coming out soon. But they found some funders that were interested, after listening to some of the sessions, to be able to look at whether and how evolutionary rescue can help corals adapt to rapidly changing climate. And it looks like they're putting a package of funding together to come out with a challenge in the near future. So thanks to the exciting work going on here, we already have funders coming forward to really work on that knowledge to action piece. I know a lot of you have been interested in asking a little bit about the Leader Summit, and I'd like to introduce my long-term friend and colleague, Noah Ideong, the past speaker of the Palau National Congress, who was instrumental in coming forward with this idea on just a couple of big points that came out from the Leader Summit. We will be posting the documents online so everybody can see it, but if I could ask Noah to report out on behalf of the leaders.
Thank you. Thank you very much, and thank you, Bob. You know, I'm ready for a six-pack, so I really want to get over this, and I'm glad that Bob is the last speaker. But I have here the document signed by the leaders. It's President of Palau, President of Federal States of Micronesia, and President of the Republic of the Marshall Islands. And so I'm not going to read it. It'll be available. But what I'll do, I really wanted to thank everyone on behalf of the leaders. I was, unfortunately, I was designated to report on the progress of the meeting. So first off, let me take this opportunity to thank Bob and ICRS and everybody for inviting the leaders from the three nations. They came with their department heads that are responsible for coral reefs and fisheries. And so we did meet and we interacted with a group of scientists and resource people. And we looked at 5 key issues. And there was a very, very good exchange between the scientists and the leaders and resource people. And after that, the document was drafted and signed before they left. And I have to say that it scares me that the leaders have actually made this statement and signed it, because now it's going to fall on Glenn Joseph from the Marshall Islands, Marion Henry from the Federal State of Micronesia, and Numi Isangaba, the minister from Pollock, to make it work. And so it's a good thing it's 4 years, Bob, because we can pace ourselves, because it's so daunting. And the leaders came, and I will tell you that the leaders, the hope may be slim, but they're working really hard at international level. COP meetings, everywhere there's opportunity, they will be pushing the agenda for coral reefs and small island. And the reason they left early is because they went to Palau to meet on the SIS, where they will report this to them also and encourage other small islands to follow suit. So the document is here. It's pretty straightforward. It's got 5 bullets, really talking about cooperation, talking about building connections, building resilience, and committing themselves to working. And that, therefore, I think it's going to be a lot of hard work. So for the future, let me say that the leaders look at it as a generalized islands, coral reefs, but the delivery system should go all the way to each island, where the practical application of the knowledge, the solutions can be made at the community level, at the state level, so that it can progress. Continuing to generalize, you know, it'll be nice, you can have nice words and keep coming up with nice words, but unless you go to the ground where the needs are and where things can work, you know, it will be continuing to, the leaders saw the need for urgent action, and they made bold commitments, and they committed their staff and their department heads to put it all together. So let me, I know we have a lot of other participants from Micronesia, Palau, and RMI. So I know Dr. Patrick DeLay is here from Palau Community College, who's been really pushing hard with the education on the ground, moving that up. And we have Dr. Golbo from Palau International Reef Center here, also been working on capacity development and working on the ground with partners. So this week, I'm very glad to see that we had a really good meeting of the mind. We saw each other, we got introduced to both sides of the table, and we talked to each other, and I think the next 4 years we'll see that the results will come. And so I really thank you. So on behalf of all the participants from Palau, FSM, and Marshall Islands, and all our friends, I really want to thank you. Thank you, Bob, and thank you for inviting the leaders to be participants here. And I want to see 4 years from now that we can be reporting a lot more progress then. So thank you so much.
Thank you. My name is Glenn Joseph from the Marshall Islands. On behalf of our leaders and to you all scientists, as managers, we hear you and we will act. And with that commitment, they also want to show their appreciation to Bob and others that work tirelessly behind the scene to make this happen. One more time.
And so in response, there were about 15 or 16, and when I say scientists, it's in the broadest sense. So biophysical scientists, we had the social scientists involved, we had people from the law side, because science is not necessarily the limiting factor, it's the political will to move the science forward. And as Noah just said, that the commitment is there from the presidents. So there is a companion document that came out from the scientists that were participating that will go out to the International Society for Reef Studies and to Ruth, and she signed it as well. That there were 4 things that we've committed to as scientists in moving forward, again, from the science to policy and the knowledge to action. And they were asking for advice. We've committed to providing technical assistance. We've committed to capacity development, and most of all, action. So I thank the members of the Leaders Summit. I thank NOAA, obviously the 3 presidents and all the wonderful staff that were there. And we definitely have our work cut out, but we have a willing partnership that we're really hoping we'll be building on. And it won't take 4 years to be able to get us where we need to go. So thank you, NOAA. Okay, so last lap. I'd like to invite Rupert to come up for the awards. And as he's coming up here, I just have an announcement that the Coral Reef Restoration Roundtable will start 10 minutes after we end, instead of at 6:30 in room 307 AB. So Rupert, if you and Ruth can come up for a very nice part. And I'd like to certainly acknowledge Joanne Leung for taking on the very large task of running a very difficult effort of being able to recognize the best of the student papers.
Okay, well, as corresponding secretary of the Society, it's my duty and pleasure to announce the winners of the student awards that ISRS is offering. Before I announce the winners, I would just like to say something about the prizes, because a number of authors and donors have been extraordinarily generous in donating large publications and glossy publications. And as a result, we're actually able to give 8 prizes, each of which consists of multiple items. Each winner will get a copy of either the Coral Finder or the Reef Finder that Russ Kelly has so beautifully put together in recent years, and it's proving incredibly popular. And in addition, they can select from one of a number of tomes that are not inexpensive. There's the Diseases of Coral that Cheryl Woodley and Jim Porter have recently produced, published by Wiley. There's Walt Goldberg's book on the biology of reefs and reef organisms that's rapidly becoming a standard textbook, and Chuck Birkeland's Coral Reefs in the Anthropocene. So there are a few copies of those available for them to choose between. In addition, each lucky winner will get an ISR t-shirt, a polo neck shirt, a dive mask strap, and 2 free years membership, plus access to 2 iDive units. These are new bits of kit that you can house an iPad in underwater and make notes as you're going along. It's technically more difficult than you might imagine, worth looking up on the website. And also we've been offered 2 free papers in Frontiers of Marine Science for whichever among the 8 winners manages to produce a manuscript first. So to go to the winners, I don't know if any of them will be here. We're not going to give the prizes actually up here, but we would like to acknowledge them. And if they're here, if they'd like to come up and be congratulated, we didn't want to try on all the t-shirts on stage. We thought that might delay proceedings. So the winners of the Best Presentation Awards are Judy Lowe from the ARC Centre of Excellence for Coral Reefs in Australia, Megan Morikawa from Stanford University, Michael Sturtevant from Florida Atlantic University, and John Whitney from the University of Hawaii at Manoa. Are any of you 4 guys actually here? If you'd like to come up, we'd love to say well done indeed. The others may already have gone off in search of their 6-packs, but look at it. Yeah. Can I just explain that, in fact, we have the awards actually at the back of the room, so if you could hang on for just a few more minutes, then we'll sort you out with what you need. Okay? So well done both. Thanks very much. And the winners of the best posters are Martin de Brouwer from Curtin University in Australia, Jeffrey Munar from the Marine Science Institute in the University of the Philippines, Zoe Sims from Princeton University, and Austin Young from Shanghai American School in Shanghai, China. So if any of you are here, please do come up. And any of the four of you here? Let me just say, in concluding, that the judging panel actually assessed over 450 entrants during the course of this conference, and the work was very consciously done by a panel led by Joanne Young, and we're very grateful to that panel for taking on that enormous task. There were a very large number of very excellent presentations from students and posters, and really there's a bit of luck in the draw here on who the winners are. So it was a very narrow margin. So congratulations to the winners, and thank you very much to the donors.
So I'm here now to say thank you to everybody for an amazing meeting. And we have so much to think about as a society. I mean, the take-homes I have from people are that we need to facilitate networks, be the networker of networks, and convene the community in a different way. And we need to be engineering ways to better communicate our science to many different places, tune our messages, come up with conversation points that actually represent the majority opinions from within our society. But one of the things that's always part of this conversation at this point is what's going to happen and what do we do in 2020. We really take the point that was made earlier about maybe convening more frequent meetings, and maybe those meetings could be in regional hubs, representing regional mini societies that fall under our umbrella. We don't know what that's going to look like. For the 2020, we have two potential locations. Neither of these are definitive settings for the 2020 meeting, but Japan and France are the two places that are really stepping forward to talk about how we might develop a bid. And I think all of the points that have been raised, there was very interesting issues raised about how we could improve the conference. These are all going to be part of a debriefing from this particular meeting, and then thinking about how to move some of the best ideas that have been put to the table forward to try and implement new things in the 2020 meeting. And so keep those ideas coming. If we, the convening over lunchtime at places that would convene scientists who are interested in a particular discipline or a particular problem. So there's many, many exciting things that have already been fed back to us. And as I said when we opened, we want to try and make our society more relevant and more serviceable and better representing what we as a community want to happen and what we're doing. We can communicate from within our society in a way that we have potentially not done before. So I'm very excited about what the next couple of years will bring for me as president, and as I step down, moving that responsibility along to another president. And I just want to say thank you for an absolutely fantastic meeting, incredibly positive and solution-oriented. It's been a really exciting week for me, and I hope it has been for you. Thank you.
So thank you all for what's been an outstanding week, and for the force of the scientific community to come together with a real pledge for trying to do the best we can to ensure that there's a legacy of reefs for the future. Helen Schneider, LeMay, and Linda West were incredibly valuable in pulling it forward. And I'd like to end by inviting a colleague of mine, Kupuna, highly respected elder who has a long history of caring for the Aina, caring for the ocean, Saul Koho Halahala, who will give us a goodbye chant.
Well, mahalo, Bob. And if I can, I'd like you to all stand and reach out to the people that are closest to you and join their hands. We began this wonderful symposium with a wonderful welcome with Kalani and setting the foundation for these discussions over the past week. And I wanted to thank Kalani for offering the foundation and the Kumulipo as where we as a people have begun and see the manifestation of many things. And I hope that you've all come to know a little bit more about our home, Hawaii, and that we can continue to move forward. So I would like to end, I think, appropriately by closing with another chant. And I would like to say a little bit about the chant. It is one that was written by one of our well-known kupuna, and her name is Kavena Pukui. And the chant talks about coming to an island home and then learning a little bit about the place that she visits. She gets to be with the people of our island. She gets to live in a home that's surrounded by the winds that cool her in the time that she stays on an island. She's taken to the highest part of the mountain. In this case, the mountain is Lana'ihale. And then she's introduced to some of the beautiful endemic plants of our islands. And in this case, she says the beautiful na'u, which is our Hawaiian endemic gardenia, and its subtlety and its scent. And then she also becomes familiar with the beautiful vines of the Mailelau Li'ili'i of the island of Lanai. And then she's taken to other parts of the island that are looking at the seashore there at Ka'a. But she says, of all of the things that I've learned coming to a small island, the thing that I am most taken by was the hospitality of the people. And so she says, in honor of the people who have hosted me and who have taken care of me, who have shared this island and the smells and the scents and the people, I want to give tribute to the people, and I want to elevate them and honor them for that kind of hospitality. And I hope for the time that all of you have had here on Oahu, and that the time that you will spend in our islands as you visit, that you will be also touched by that aloha and that spirit of hospitality. And so in that light, I would like to offer this oli to you. Hano, hano kei kena ia oe, e na nai, aina o nauna me te aloha. Ai ka olu o ni vai, I ka pā ahe ka makani. Nani nanai ta mahali, yeah. Nani nanai ta mahali, yeah. Kau aku na matai te tuahiwi, I tohu hali o nanai hale. Me ta puanau, ko ho'ihi, me ta maile lauli o kanepu. Nani nanai ta mahali, yeah. Nani nanai ta mahali, yeah. Ina ke aloha no, e na nai, ai na ho o hihi a ka puwai, ai I laila ka loko mai ka inui, o na maka hoki paloa. Nani nani kaui ka hano, nani nani kaui ka hano, aloha e, aloha e. Aloha and ahui ho until we meet again.